as we start to go through these different processes, like meiosis, the stages of meiosis, we're going to compare and contrast between what's going on in mitosis. So in prophase one, we're going to see some similar features as to mitosis. We are going to go take that relaxed chromatin and we're going to pack it up tight into our chromosomes. So we're going to go from spaghetti to Cheeto form in our DNA. We also need to get rid of that nuclear envelope so that we can move all of the chromosomes around in the cell. Okay, that's the same that we saw in mitosis. The thing that's different that we see in prophase one that we did not see in mitosis is we are going to have these homologous chromosomes come and associate with each other temporarily during this process called synapsis. Okay, and when those homologous chromosomes are in close proximity to each other, we are going to see them kind of get tangled up, and we're going to see them actually swap portions of their non-sister chromatids and create a novel combination of genes. Okay, so crossing over is our second source of genetic variation during the process of sexual reproduction. Crossing over is the exchange of genetic material between non-sister chromatids that gives us a novel genetic combination, right? This doesn't exist anywhere else in the universe. So if we look at this again, our homologous chromosomes that are replicated, we have our sister chromatids, right? So this is the exact copy of this portion of the chromosome, but this portion of this chromosome is not necessarily the same as this portion of that chromosome, okay? So we call those non-sister chromatids, right? These two are sisters, these two are sisters, but this one and this one are not sister chromatids, okay? So what those non-sister chromatids are going to get tangled up, and we're actually going to see part of that break off and exchange places with portions of the other non-sister chromatid. Again, the point of this is to shuffle up those genes, create novel combinations. All right, so that was prophase one. In metaphase one, we're still lining stuff up along the equatorial plane, but instead of lining up, right, each of the chromosomes, right, so that our sister chromatids can separate, we're actually lining up pairs of homologous chromosomes. So when we separate, we're going to be separating those homochromos, okay? And the important part of metaphase one is that as those homologous chromosomes are lining up along that equatorial plane, they're going to line up independent of each other. So if we get the orange marker from mom and the blue marker from dad, each set of homologous chromosomes can arrange themselves in either orientation. It doesn't matter. Okay? They're independent of each other. So that is going to give us, again, when those cells separate, we're going to get a different gene combination in the daughter cells. Okay, so that's our third source of genetic variation is independent assortment, and it occurs in metaphase one. The number of chromosomes that you have is going to lead to more unique and distinct gene combinations. So for humans that have 23 chromosomes, there are over 8 million unique combinations that our chromosomes can line up and separate. So that's really quite incredible amount of genetic variability that can arise just through this one stage of process of meiosis. All right, so in our anaphase one, we are going to, again, as I kind of alluded to, kind of played my hand early, we're going to separate the homologous chromosomes. So each homologous chromosome is going to stay paired as their sister chromatids, right? We're still in double Cheeto form. We're just separating the homologous chromosomes. And that is the point during the process of meiosis that these things are considered to be haploid. So we did prophase 1, we did crossing over. Metaphase 1, we did independent assortment. Anaphase 1, we separated homologous chromosomes and we became haploid. And telophase 1, this looks just the same as it did in mitosis. Okay, we're going to have the chromosomes relax back into the chromatid form, so going from Cheeto to spaghetti form. We're going to rebuild that nuclear envelope, and cytokinesis is going to occur at the same time. So at the end of meiosis 1, we end up with 2 cells.